Welcome to PCR primers, the DNA amplification revolution. Today, we're diving into the origin story of a technique that fundamentally changed molecular biology. Sam, before we had PCR, what was DNA analysis like for researchers?
That's a great question, Alex. Before PCR, working with DNA was incredibly challenging, especially when you only had trace amounts. Imagine trying to study a specific gene or diagnose a condition from a tiny sample. It was often next to impossible to get enough material. Researchers were limited by the quantity of DNA they could obtain, and amplifying specific sequences was a laborious, often inefficient process. The precision and speed we now take for granted simply didn't exist.
So, a major bottleneck. And then, everything changed with a eureka moment, right? Tell us about Kary Mullis.
Absolutely. The change agent was indeed Kary Mullis, an American biochemist. The story goes that in 1983, while driving on a moonlit night in Mendocino County, Mullis had this profound revelation. He was working at Cetus Corporation at the time, already focused on DNA synthesis, but he envisioned something entirely new, a cyclical process for DNA amplification. This wasn't just an incremental improvement, it was a fundamental shift, an idea that had remarkably eluded others for decades.
A cyclical process for DNA amplification. That sounds like the core of PCR. How did primers fit into this initial concept?
Primers were, and remain, absolutely crucial to Mullis's invention. At their heart, PCR primers are short, single-stranded DNA fragments. They typically range from 18 to 25 nucleotides in length. In Mullis's vision, these primers would serve as the essential starting points for DNA synthesis. Without them, the enzyme responsible for building new DNA strands, DNA polymerase, simply cannot initiate the process. They define the precise region of the DNA template that will be amplified, ensuring both the specificity and efficiency of the reaction.
So these small DNA fragments are like signposts, telling the DNA polymerase exactly where to start copying. And this wasn't just about making more DNA, but specific DNA, right?
Precisely. The genius of PCR, enabled by these primers, is its ability to rapidly and exponentially amplify specific DNA sequences from even trace amounts of template DNA. Before PCR, isolating a specific gene from a complex genome was incredibly difficult. Mullis's invention allowed researchers to target and multiply a desired segment, producing countless copies of a target gene or DNA segment in a matter of hours. This level of specificity and amplification was truly revolutionary.
It sounds like a giant leap forward, and its impact was recognized globally, wasn't it?
Indeed, the significance of PCR was immense and undeniable. Just 10 years after his invention, in 1993, Kary Mullis was awarded a share of the Nobel Prize in Chemistry for his groundbreaking work. His invention, heavily reliant on well-designed primers, became indispensable across various fields. Think medical diagnostics, forensic science, genetic research, and biotechnology. It truly opened up new avenues for discovery and application.
It's incredible how one idea, conceived on a drive, could have such a profound and lasting effect on science and medicine. So, from that 1983 eureka moment, PCR quickly became a cornerstone technique. What was it like in those very early days of running a PCR experiment?
In its initial form, while revolutionary in concept, PCR was still quite a manual and labor-intensive process. The method requires cycles of heating and cooling. At the high temperatures needed to separate DNA strands, the DNA polymerase enzyme would denature, meaning it would become inactive. So after each high-temperature step, researchers had to manually add fresh enzyme. It was a tedious cycle, but the promise of endless specific DNA amplification kept them going. It was clear that to truly automate and streamline the process for widespread adoption, a major hurdle remained.
Manually adding enzyme after every cycle sounds like a logistical nightmare, making it very laborious. It's hard to imagine something so fundamental still having such a significant practical challenge. So the concept was brilliant, but the execution needed refinement. What came next to address this?
That's exactly right, Alex. The manual enzyme replenishment was the biggest practical bottleneck. For PCR to move from a specialized lab technique to a widely adopted, efficient, and automated process, something fundamental about the enzyme itself needed to change. The search was on for a DNA polymerase that could withstand those high denaturation temperatures, eliminating the need for constant manual intervention. And that search eventually led to some rather surprising places, specifically to extreme environments like hot springs.
Intriguing. So the next step in this revolution involved looking to nature's extremes to find an enzyme that could handle the heat, transforming PCR into the automated wonder we know today. We'll explore that breakthrough, the discovery of Taq polymerase, and its journey from hot springs to widespread adoption in our next episode, From Hot Springs to Automation, the Taq polymerase breakthrough. Welcome back to PCR Primers, the DNA amplification revolution. In our last episode, we delved into the incredible eureka moment of Kary Mullis in 1983, when he conceived the core idea of PCR while driving. He envisioned a cyclical process for amplifying DNA, a concept that fundamentally changed molecular biology. But having a brilliant idea is one thing, making it practical for everyday lab use is another. Sam, what were the initial hurdles that Mullis and other early researchers faced right after the invention of PCR?
That's a fantastic point, Alex. Kary Mullis's invention of PCR in 1983 was indeed revolutionary. However, the initial iterations of PCR were incredibly laborious and time-consuming. The fundamental challenge lay with the DNA polymerase, the enzyme responsible for synthesizing new DNA strands. PCR involves cycles of heating to separate DNA strands, a process called denaturation, and then cooling for primers to anneal and polymerase to extend. The problem was that the DNA polymerase available at the time, typically from E. coli, couldn't withstand the high temperatures required for the denaturation step. Every single cycle after the DNA strands were separated by heat, the enzyme would be destroyed.
So, if the enzyme was destroyed in each cycle, what did researchers have to do to keep the reaction going? That sounds incredibly frustrating. So, what was the breakthrough that transformed PCR from this laborious manual process into the automated, efficient technique we know today? And where did this solution come from?
The breakthrough came in 1985 with the introduction of Taq polymerase. This was a critical advancement. Researchers discovered a heat-stable DNA polymerase isolated from a thermophilic bacterium called Thermus aquaticus. As its name suggests, Thermus aquaticus thrives in extremely hot environments, such as hot springs, meaning its enzymes are naturally resistant to high temperatures.
So an enzyme from a bacterium living in hot springs was the key to unlocking PCR's full potential. That's quite a journey for a protein.
Precisely. Taq polymerase could endure the high denaturation temperatures, typically around 95 degrees Celsius, without losing its activity. This meant that researchers no longer needed to manually replenish the enzyme in each cycle. Once added at the beginning of the reaction, Taq polymerase would continue to function throughout all the amplification cycles. This single discovery fundamentally transformed PCR. It eliminated the need for constant human intervention, making the entire process automated, much faster, and significantly more cost-effective.
And I imagine this automation paved the way for dedicated machinery.
Absolutely. With a heat-stable enzyme, the path was clear for automation. Commercial automated PCR thermal cyclers, which precisely regulate the temperature cycles for denaturation, annealing, and extension, first became available in 1988. These machines could run a PCR experiment unattended for hours, generating countless copies of a target DNA segment. This combination of Taq polymerase and automated thermal cyclers made PCR accessible to virtually any molecular biology lab, solidifying its role as an indispensable tool. It's no exaggeration to say that without Taq polymerase, PCR would likely not have achieved its ubiquitous status in medical diagnostics, forensic science, genetic research, and biotechnology.
It truly sounds like a pivotal moment, taking a brilliant concept and making it a practical reality for countless applications, from identifying infectious agents to solving cold cases. And Mullis even shared the Nobel Prize in Chemistry in 1993, a recognition that, in part, surely acknowledged the technique's eventual widespread utility, thanks to these advancements.
Indeed, the impact was global and profound. The ability to amplify specific DNA sequences exponentially, quickly, and affordably opened up entirely new avenues of research and application. It allowed for the production of countless copies of a target gene or DNA segment in a matter of hours, making it possible to work with even trace amounts of template DNA. It's a testament to how a single innovation can drastically change an entire scientific field.
What an incredible journey, from a hot spring bacterium to revolutionizing a lab technique. So, with the enzyme problem solved and automation in place, PCR seemed unstoppable. But even with these breakthroughs, the specificity and efficiency of the reaction still heavily rely on one crucial component we mentioned in our last episode, the primers. What kind of challenges do researchers face when designing these short DNA fragments to ensure the reaction works perfectly every time?
That's a fantastic question, Alex, and it brings us right to the heart of our next episode. While Taq polymerase solved the automation puzzle, poorly designed primers can still wreak havoc, leading to issues like nonspecific binding, low amplification efficiency, or even false positive results. Getting primer design just right involves a delicate balance of length, GC content, and sequence uniqueness. Join us next time as we uncover the unseen saboteurs, primer dimers, and nonspecific binding, and explore the sophisticated tools developed to mitigate these critical challenges.
Welcome back to PCR Primers, the DNA amplification revolution. In our last episode, we explored the incredible breakthrough of Taq polymerase, which transformed PCR into an automated, efficient technique. Today, Sam, we're diving into what makes PCR so precise, and what can go wrong if we don't pay close attention to the details of its critical components, the primers.
That's right, Alex. PCR is powerful, but its success hinges almost entirely on well-designed primers. These short, single-stranded DNA fragments, typically 18 to 25 nucleotides in length, are the starting points for DNA synthesis. They define the specific region of the DNA template that will be amplified, ensuring the reaction's specificity and efficiency. Without them, DNA polymerase can't even begin its work.
So they're not just any random DNA snippets then. What happens if they're not designed correctly? Are there common pitfalls that researchers encounter?
Absolutely. Poorly designed primers are, as our episode title suggests, the unseen saboteurs of a PCR experiment. The most common pitfalls are primer-dimer formation and nonspecific binding. Primer dimers occur when primers bind to each other instead of the target DNA, forming an unwanted product. Nonspecific binding, on the other hand, means the primers bind to incorrect regions of the template DNA, leading to amplification of unintended sequences.
That sounds like it could throw off an entire experiment, resulting in false positives or just no useful results at all.
Precisely. Both issues can lead to low amplification efficiency and inaccurate results. It's a delicate balance, and several factors are crucial for optimal primer design. For instance, the length of the primers. They're generally 18 to 25 nucleotides long. Then there's the melting temperature, or TM. The optimal TM for PCR primers typically falls between 58 degrees Celsius and 60 degrees Celsius, and crucially, the TM of both the forward and reverse primers in a pair should ideally be within 1 degree Celsius of each other.
Only 1 degree Celsius difference. That's incredibly precise. What other factors are involved?
Another vital factor is the GC content, which is the percentage of guanine and cytosine bases. This should ideally be between 40% and 60% for optimal binding strength. Too low, and the primers might not bind strongly enough. Too high, and they might bind too strongly or form secondary structures, leading to primer dimers or nonspecific binding. Also, the optimal length for the amplified DNA fragment, or amplicon, for efficient PCR is usually between 50 to 150 base pairs, though it can range from a few dozen to several thousand base pairs.
It sounds like a complex puzzle to solve for every new target DNA sequence. How do molecular biologists manage all these parameters to ensure their primers are perfect?
That's where technology steps in. Initially, it was a lot of manual calculation and trial and error, but today, sophisticated computational tools are indispensable. Primer design tools like NCBI's Primer Blast are widely used. They combine primer design features with specificity checks against genomic databases. This helps researchers avoid non-target amplification by ensuring their chosen primers are unique to their desired sequence and won't bind elsewhere in the genome.
So these tools effectively act as a digital safety net, pre-screening potential primers for those.
Exactly. They significantly reduce the chances of encountering those unseen saboteurs. Companies like Co-Diagnostics Inc. have even developed patented approaches like co-primers technology to increase PCR accuracy and specificity by explicitly reducing primer-dimer formation. And specialized companies like Zinke are committed to providing high-purity primers for efficient molecular biology applications, knowing how foundational they are.
It's fascinating to realize that for PCR to work its magic, there's this entire layer of intricate design and optimization happening behind the scenes, ensuring the building blocks, the primers, are precisely crafted. It really underscores the ingenuity involved in making a technique like PCR so reliable.
Indeed. The precision in primer design is what allows PCR to be such a versatile and accurate tool. Without it, the revolution Kary Mullis started in 1983, for which he shared the Nobel Prize in Chemistry in 1993, wouldn't have reached its full potential. The heat-stable Taq polymerase, introduced in 1985, automated the process, and by 1988, commercial automated PCR thermal cyclers were available. But none of this would matter without effectively designed primers.
It makes you appreciate the engineering behind the biology. From those initial eureka moments to overcoming technical hurdles, the journey of PCR is truly remarkable. And it's clear that mastering primer design is absolutely fundamental to harnessing the full power of DNA amplification.
It is. And this meticulous design isn't just for theoretical research. These precisely engineered primers are critical for making a tangible difference in the world, addressing pressing issues and solving complex problems that touch our daily lives.
That's a perfect lead into our next episode. Join us next time on PCR primers, the DNA amplification revolution, as we explore how this technique, once optimized with specific primers, moved swiftly from research labs to solve real-world problems. You won't want to miss it. Welcome back to PCR primers, the DNA amplification revolution. We've explored the groundbreaking invention of PCR by Kary Mullis in 1983 and the pivotal role of Taq polymerase in automating the process. Today, we're moving beyond the lab bench to see how this revolutionary technology, powered by those essential PCR primers, has fundamentally impacted the real world. Sam, it's incredible how a technique conceived in a car can have such far-reaching consequences, isn't it?
Absolutely, Alex. It truly is. PCR primers, those short single-stranded DNA fragments, typically 18 to 25 nucleotides in length, are the unsung heroes that make PCR so powerful. They define the precise region of the DNA template that will be amplified, ensuring the specificity and efficiency of the reaction. Without them, the DNA polymerase cannot even begin synthesizing new strands. This specificity is exactly what makes PCR so indispensable across diverse fields, turning it from an academic concept into a critical tool for solving real-world problems.
So let's dive into some of those real-world applications. Where do we see PCR having the most significant impact today, thanks to the precision offered by well-designed primers? One of the most immediate and profound impacts has been in medical diagnostics, particularly in identifying infectious agents. Think about recent global health challenges like the COVID-19 pandemic. PCR played a pivotal role. The ability to rapidly amplify specific DNA or RNA sequences from even trace amounts of a pathogen's genetic material meant we could quickly and accurately detect infections. This rapid identification is crucial for public health responses, allowing for timely isolation, treatment, and containment strategies. Before PCR, such rapid and sensitive detection methods were largely unavailable, making diagnosis much slower and more challenging. That's a fantastic example. The speed and accuracy must have been game changers during a public health crisis. What about other areas where precision DNA amplification has been transformative?
Another area where PCR has made an astonishing difference is in forensic science. The ability to amplify specific DNA sequences from minute DNA samples found at crime scenes has revolutionized how investigators solve cold cases and identify individuals. Imagine a tiny drop of blood or a few skin cells left behind. With PCR, scientists can amplify specific regions of that DNA to generate a genetic profile. This has led to breakthroughs in cases that were once thought unsolvable, bringing justice to victims and closure to families. It's a powerful testament to how a technique relying on carefully selected primers can have such a profound societal impact.
Solving cold cases, that's incredibly impactful. It speaks to the sensitivity of the technique. And I imagine it's also crucial in genetic research and diagnostics, right?
Absolutely. In genetic research, PCR, guided by those specific primers, allows scientists to isolate and study particular genes or DNA segments. This has deepened our understanding of genetic diseases, evolutionary biology, and gene function. More directly in diagnostics, it's now a standard tool for diagnosing genetic disorders. For example, if we suspect a patient has a particular genetic mutation, PCR can be used to amplify the specific region of their DNA where that mutation is known to occur. This allows for early and accurate diagnosis, which is critical for medical management and genetic counseling. The optimal length for these amplified DNA fragments, or amplicons, for efficient PCR is usually between 50 to 150 base pairs, although it can range from a few dozen to several thousand. This flexibility allows for targeting a wide range of genetic markers.
So, from infectious diseases to crime scenes to genetic predispositions, PCR, with its core reliance on primers, has truly moved beyond the test tube to become an essential part of our modern world. It's a testament to Kary Mullis's original eureka moment in 1983.
Precisely, Alex. And it's important to remember that the widespread adoption and automation of PCR wouldn't have been possible without key innovations, like the introduction of Taq polymerase in 1985, which eliminated the need to add fresh enzyme in each cycle, or the commercial availability of automated PCR thermocyclings in 1988. These advancements, coupled with the meticulous design of primers, have transformed PCR into the versatile and indispensable technique we know today. Its societal impact continues to grow, touching countless lives in ways Mullis might not have fully envisioned when he conceived the idea. A truly revolutionary technology. Thank you, Sam, for illuminating the incredible real-world impacts of PCR and its essential primers. Join us next time as we continue our journey through the DNA amplification revolution.